So we've been talking about proteins and how we have a polypeptide chain, basically the primary structure, and how that primary structure starts to fold into the secondary structure and the tertiary structure, and then finally maybe even the quaternary structure to where you get a functional protein. If you have no idea what I'm talking about, I highly recommend you go watch our protein video. In this video, we're going to be talking about how proteins are kind of manufactured, how they're made. Now, that's actually a very, very extensive topic that we're going to be studying in AP biology, how proteins are made, because it's very important, and you're going to learn all about that in our genetics unit. But this video is going to be kind of an introduction to that. And while we're also teaching you how proteins are made in this video, we're also going to be covering the last type of biomolecules, the last category, which are nucleic acids. So let's get right into it. Okay, so we said that we have let me write this down. We have the primary structure for a protein, which is, remember, the amino acid sequence, right? So that's just the long chain of amino acids. Now, what determines what is going to be the primary structure for that protein? Okay, so that comes from a gene. Okay, so you can think of the gene as the information which is going to tell the cell what the primary structure for the amino acid is. So once you get the primary structure for the protein, then the hydrophobic interaction is going to occur. Let me write that down. Hydrophobic interaction. And then with the hydrophobic interaction, you're going to go from the primary structure to the secondary, tertiary, all the way up to the functional protein. Let me write that in a different color. Functional protein. Okay, so these genes here, basically the things which are let's say this is information. Okay, you can think of this like kind of like within an office, all the important paperwork or something like that, So that's the information, kind of the brain of the cell, is stored in the gene, and nucleic acids make up these genes, okay? So a lot of nucleic acids make up these genes, all right? So I hope you understand the sequence here. Let me explain that. Nucleic acids make up the gene. The gene is the information which codes for the primary structure of the protein, okay? And we're going to be talking about this. Just give me a second. I'll explain how this happens. And that primary structure is going to go through the hydrophobic interaction in the cell, and then through the hydrophobic action, which we talked about, this whole process, primary to secondary to tertiary structure, we're going to get the functional protein, okay? If you don't understand what all of this mess is, then I think you need to go watch the protein video. I'm going to write a note for you here. Watch protein video, okay? Which we have made. Okay, it's in the biomolecules playlist, and you can also access it on the website. Okay, so that's when I say this bullet point here, that the nucleic acids carry genetic information, essentially what I'm saying is that the information is stored in the gene, and that's basically this whole structure here, okay? So there's basically two types of nucleic acids. There are DNA, which carries information, and then there's RNA. So let me give you an analogy. Let's say you have a Word document, or just something you've typed on your computer, okay? So let's like just something like a book report, okay? So I'm just going to say document, okay? And you want to print this document out. So when you print this out, what you have to do is you have to go to File, Print, and you have to print that, right? So that converts it to a software, okay? So the software is going to take the document, and it's going to convert it into information which can relay to the printer, okay? And then once it can get to the printer is going to change it, and it's going to print it to, the printer is going to be the place where the actual, sheet of paper, the paper is going to be made, right? This is, a pretty common sequence. In the same way, I'm going to use this analogy to explain how proteins are made. So you have the genes, or the nucleic acids, so that would be the document, nucleic acids, specifically the DNA, okay? It's called DNA. DNA is the genetic information, so this is the DNA, okay? Remember that DNA stands for deoxyribonucleic acid, okay? Kind of tough to remember, but you need to memorize that, okay? And then this DNA is going to be converted into information, which this whole process we're going to be diving in way more later in this course, but this is just what you need to know right now. So this DNA, the nucleic acids, are going to be converted into information which can be relayed to the printer, So like that is done by the RNA, Okay, so you go from DNA to RNA. You can think of it just like the software. Then the RNA is going to be taken to something called a ribosome. Okay, in the same sense that you have a printer making the paper, you have a ribosome, and the ribosome is going to create the protein. Okay, that's the factory. In the same way, and it's, when I'm talking about the protein, I'm talking about just the primary structure. Okay, it's not going to make the protein into functional protein. What makes the protein a functional protein is the hydrophobic interaction, which we talked about in previous videos. But it's just going to make the primary structure for the protein. Okay, I hope you understand the sequence. So again, nucleic acids are the document, then it's converted, the software, kind of like how a software, when you hit Control P, or file print, that's what the RNA does. It relays the information from the document to the printer, essentially DNA to ribosomes. Then the ribosome is the printer. It makes it takes that information and actually physically makes it into a protein. Okay, so we got that cleared up. Now, let's talk about the monomers for nucleic acids. So just like how all the other biomolecules have monomers, nucleic acids also have monomers. They're called nucleotides. Okay, nucleotides. Now, what does a nucleotide consist of? We've talked about functional groups, so it's going to be important that you see this again. So we have the phosphate group here. This is a phosphate group if you look here. And then we have that bonded to this kind of like a sugar here. This is a sugar. Okay, and then attached to the sugar you have what is called a nitrogenous base. So let me label that. So this is the phosphate group. Okay, this is the sugar, and then this is the nitrogenous base. Okay, so they're kind of, in all nucleotides they're connected like this. Okay, so in DNA, sugar is deoxyribose. Okay, so just you have to memorize the difference between DNA and RNA. So in DNA, the sugar is deoxyribose. Okay, and the nitrogenous bases are adenine, thymine, cytosine, and guanine. Okay, this is something you probably learned in pre-AP, but if you forgot, this is something you need to remember. In DNA, the sugar is deoxyribose, so the sugar right here is deoxyribose, and then the nitrogenous bases can either be adenine, thymine, cytosine, or guanine. So the nitrogenous bases right here can either be adenine, thymine, cytosine, or guanine. Okay. In comparison, in RNA, the sugar is ribose. Okay, and it's a little different from deoxyribose. You don't really need to know how. Just make sure that you know that the sugar here is ribose, and the nitrogenous bases could either be adenine, uracil, guanine, and cytosine. So instead of having thymine in DNA, you have uracil instead, the nitrogenous basis right here. Okay, make sure you memorize that. Very important. Everything else is the same, except that thymine is in RNA is actually uracil. Instead of thymine, you have uracil. Okay, and this is also important here. Based on the nitrogenous bases, they're classified into two different groups, the pyrimidines and the purines. Okay, in pyrimidines you have a one carbon ring. Okay, so these are cytosine, thymine, and uracil. Kind of like you have just one carbon ring right here. In purines you have adenine and thymine, and there's two. Adenine and thymine are purines. Okay, and those are two carbon rings. Let me show you a picture here. So compare that. This would be the pyrimidines. This would be an example of a pyrimidine, while this is a purine because there's two carbon rings fused together. Okay, and again, the pyrimidines are cytosine, thymine, and uracil, one carbon ring, and the purines are adenine and thymine. Okay, a lot of things you have to take in. Make sure you understand this and you memorize it. Don't just watch this video and then move on. Okay, so those are the monomers. Now let's talk about how the polymers of nucleic acids come together. Okay, so the difference between DNA and RNA is that DNA forms what is called a double helix. Okay, kind of down here. So this is DNA. You can kind of see that here, right? Because you have a double helix. You have one strand which is going this way. Let me see if I can highlight that. One strand which is going this way. That's the one. And then you have another strand which is going this way. Okay. Meanwhile, RNA is single-stranded. Okay, there's just one row like this. So this is RNA. So you can see that there's only one chain like this. Okay, so the way this works is that adjacent nucleotides are bonded by what is called a phosphodiester linkage. A phosphodiester linkage, very important. Okay, And a phosphodiester linkage, we've been talked about all sorts of linkages through this video. I mean, through this course, you know, we talked about a ester linkage, and we talked about, I can't remember it from the top of my head, but all these linkages are created by hydrolysis. And this too, these linkages are also created by polymerization, also known as dehydration synthesis. And we have a video on polymerization and hydrolysis. You can go watch that video too. I'll make a note here. Watch the hydrolysis and the polymerization video if you don't know what that means. Running out of space here video, okay? So if you don't know what that means. And I showed you all the examples of how, you know, there is a OH group reacting with the hydrogen and they, you know, come apart to form, they leave, you know, to create a extra water molecule, and there's a single bond which is formed. And the same thing happens within nucleic acids, okay? So all these linkages here, if you look here, we had a phosphate group, and then you have a sugar, and then you have the nitrogenous base. So if you looked at all these linkages here between each group, specifically right here, these linkages right here, these are all phosphodiester linkages, okay? So, anyways, just make sure you know that. So what determines the information, right? So we said that here that the DNA is kind of like the document. It determines what's going to be made at the very end. It's the information, right? So just like how you're going to type a Word document and you're going to print it out and it has that information on the Word document, what determines the information for a protein, right? So what determines is basically within the structure of DNA here. So DNA is a double-stranded. So instead of, this is an RNA molecule, remember that. This is a DNA. I can just move here, okay? So this is a DNA molecule, right? So what determines the information of what the protein is going to be coded for, what protein is going to be made, is basically the sequence of the nitrogenous bases. So let's just say, in this example, that this is cytosine. Any yellow box is cytosine, okay? And then any green box is guanine, and then any purple box is adenine, and any orange box is thymine. So basically, depending on the sequence of these nitrogenous bases, you know, that sequence of how they come about within a DNA molecule is going to determine the information that the sequence carries, okay? Now, let's go back to this double helix, because there's a few more things that I want to mention, okay? Actually, I don't think I mentioned this here, okay? This is something I forgot to mention. So if you look at this group here, there's actually a way that the carbons are numbered, okay? So in the sugar, you have carbons here at every single point. We talked about this when we talked about rings and things like that, with how carbon can be formed rings and stuff like that. So basically, there's a carbon here, there's a carbon here, there's a carbon here, there's a carbon here, and then there's finally a carbon right here. So they're actually numbered in a way. This is called the 1' carbon. This is called the 2' carbon. This is called the 3' carbon. I've already labeled it. This is called the 4' carbon, and this is called the 5' carbon. So if you look here, you can also determine it here. So in this structure right here, you have 1, 2, 3, 4, 5. Also here, if you look carefully, 1, 2, 3, 4, 5. Now, here we have just a RNA molecule, so it's single-stranded, but in a DNA molecule, you have something called antiparallel. Okay, that means the DNA strands run opposite to each other. So if you look here, this would be maybe a 3 to 5. Okay, because you're starting with a 1, 2, 3 prime, right? And then at the very end, you end up with a 5 prime. So it's going 3 prime to 5 prime. So on the opposite side, if you look here, it kind of intertwines, right? You have one DNA strand with the red right here, and then you have another DNA strand with the blue back bone right here, right? So this red strand would go 3 3 to 5 prime. It's kind of ambiguous. It can go either way. It's either way you look at it, right? But the opposite strand is going to go 3 prime to 5 prime. So it's going to, at one side, you're going to start with the 3 prime carbon and end up with the 5 prime carbon, and then for the other strand, you're going to start with the 3 prime carbon and end up with the 5 prime carbon like this, okay? So that's what this means, that DNA runs in anti-parallel directions, okay? Now, an important thing in DNA is that adenine always bonds to thymine and cytosine always bonds to guanine, okay? Very important. If you look here, if you see here, cytosine is bonded to guanine here. Okay, if you look anywhere in this molecule, you can see that. Here we have I said any orange box is a thymine molecule, and look, since this is a thymine on this side, it's bonded to an adenine here. Here we have a cytosine, right? And it's bonded to a guanine. You can kind of see that with the colors. You're never going to see a orange mixed up with a green or a purple mixed up with a green. If there's a cytosine, it has to be bonded to a guanine and vice versa. And if there's an adenine, it has to be bonded to a thymine and vice versa. Okay? So if you get a question like this is a very common question that your teacher might want to ask, you know. If you have this basically asking if you have one strand of the DNA, let's say you have the 5 prime to 3 prime. So let's say this red thing right here, 5 prime to 3 prime, and they give you the sequence of that. Then what is going to be the opposite, okay? What is going to be the blue side, the 3 prime to 5 prime? So the way we write this is you have this. Let's do this problem. So it's going to be, first you remember that you have to write 3 prime because you're going in one direction, so you have to write the opposite direction, right? Because I said 5 prime to 3 prime. The other one was 3 prime to 5 prime. So you're going to say 3 prime dash, and then we say cytosine always bonds to guanine, right? So G. Guanine. Thymine always bonds to adenine, so A. Adenine always bonds to thymine, so T. Guanine always bonds to cytosine, so C. T, C, C, A. 5 prime. Okay. Okay, so now let's do another example here where we're given the 3 prime to 5 prime backbone, and we want to find the 5 prime to 3 prime. So let me just go ahead and erase this. So what we're given is the blue backbone. We're giving the 3 prime to 5 prime, and what we're looking for is the 5 prime to 3 prime. Okay, this is what we're looking for. Okay, sorry. So again, since this backbone is going from 3 prime to 5 prime, then the opposite backbone is going to have to go, the opposite strand is going to have to go 5 prime to 3 prime. So we start with 5 prime, and then we know that guanine always bonds to cytosine, so C. Thymine always binds to adenine, so A. Cytosine always bonds to guanine, so G. Adenine always binds to thymine, so T. Thymine always bonds to adenine, so A. And then cytosine always bonds to guanine, so G, and then 3 prime. Okay, the reason why I went through this so quickly is because this is something you should have learned from pre-AP. If you didn't, then make sure you remember this. Adenine always bonds to thymine, and cytosine always bonds to guanine, okay? So that's pretty simple. The last thing I want to mention about DNA are these 2 notes here. DNA can replicate very easily because of the way bases pair. This whole process is actually called something called DNA replication, and DNA replication is something that we're going to be discussing way down the road in molecular genetics. But just basically as a brief introduction, essentially when DNA replicates, well, why would DNA have to replicate? Okay, DNA is actually, this is something you don't know, but DNA is found within your body, or within any organism for that matter. Every single cell within that organism is going to have the same DNA. Okay, what makes a cell different is because even though the DNA is the same between cell to cell, which part of the DNA is actually going to be coded for proteins, and that's something which is kind of hard to understand, but don't really worry about it. What I'm trying to say is that whenever this DNA is replicated from cell to cell, the reason why DNA can replicate so easily is because of the way the bases pair. So what happens really is this DNA unwinds and it leaves each base strand, and you can, if you have a good intuition, you'll be able to understand this, how because when the DNA unwinds, since the base pairs are nicely in this manner. Adenine always binds to thymine, and cytosine always bonds to guanine. And if this DNA strand just comes apart like a zipper, it's going to be very, very easy for the for two DNA molecules to be created from one. And if that doesn't make sense, that's okay, because we're going to be actually showing you a video of that way down the road. You don't really have to know that for this video, but I think this is something to mention now, just so later on the road you might be like, hey, Shreyas talked about this, I already kind of know what he's talking about, okay? Also, another big thing that you need to know is that DNA plays a very big role in evolution and heredity. Organisms which are similar will always have similar DNA sequences, because think about it, DNA is the information which codes for what an organism is. It's kind of like your blueprint. So if you have organisms which are similar, they should have similar DNA. So for example, siblings will have more similar DNA than random strangers. So if you have a sibling and you did a DNA analysis, you notice that your DNA would be significantly more similar with your sibling than some random stranger that you found on the street, okay? Also, this goes from, from species to species as well. Humans have similar DNA compared to each other. If you look at a human hand, your human hand is always going to look way more similar to another human hand than like a human with a chimpanzee, right? So humans have similar DNA compared to each other than compared to other species. Okay, so basically, the more evolutionary, laterally you're related, like through evolution and heredity, the closer you're related, the similar the DNA should be. And I think that's kind of obvious, but if it was not, I just had to mention that, okay? So that's all you have to really know for nucleic acids. Nucleic acids are as simple as that.